PCH 180151 Unit 4 Bioinorganic Chemistry. This is video lecture 2. I am Dr. Devang R. Pandya from Department of Chemistry, St. Xavier's College, Autonomous, Ahmedabad. In this lecture, we are going to discuss the effect of chelation and the metal chelated complex and its application in medicine. Chelation is a type of bonding of ions and molecules to metal ions. That means metal and bigger biological group of atoms will going to form a coordinating bond and give a cyclic structure and form a chelate. That process of binding of ions and molecules to metal ion in a cyclic way is known as chelation. It involves the formation or presence of two or more separate coordinate bonds between a polydentate ligand. When I say polydentate ligand, then it has more than two donor atoms in it and create a cyclic structure with the central metal ion, and a single central atom will going to form a ring-like structure that is called chelate, and the entire process of forming this kind of bonding is known as chelation. Usually these ligands are organic compounds and are called chelants, chelators, chelating agents. Some may either use different pronunciation like chelatants, chelators, or chelating agent, but actually it is called chelating agents or sequestering agents. It is also known as sequestering agents as well, and they are polydentate ligands. That means they have more than two donor atom in it, which have ability to donate lone pair electron to a single central metal ion to form a ring-like structure known as chelate. Chelation is useful in applications such as providing nutritional supplements in chelation therapy to remove toxic metals from the body, as contrast agents in MRI scanning, in manufacturing using a homogeneous catalyst, in chemical water treatment to assist in the removal of metals, and in fertilizers. So these are the area where widely chelated compounds are used. Nutritional supplements are there, remove toxic metals from the body as chelating therapy. We can use it as contrast agent in magnetic resonance imaging scanning. It is used in manufacturing of homogeneous catalyst and in chemical treatment of the water to remove the metal, and in fertilizers as well. Chelating compounds have vital application. Now chelate in nature. How nature is going to form a chelate and that is useful. Numerous biomolecules exhibit the ability to dissolve certain metal cations. Thus, protein, polysaccharides, and polynucleic acids are excellent polydentate ligands for many metal ions. Organic compounds such as the amino acid, glutamic acid, and histidine, organic diacids such as malate, and polypeptides such as phytochelation are also a typical chelators, or we can say chelating agent to form chelate complex. In addition to these advantageous chelators, several biomolecules are specially produced to bind certain metals. Let's see in biochemistry or microbiology how chelating agents are used. Virtually all metalloenzymes features metals that are the chelated compound, usually to peptides or cofactors or prosthetic groups like porphyrin. Such chelating agents include the porphyrin rings in the hemoglobin and chlorophyll compound. Many microbial species produce water-soluble pigments that serves as chelating agents termed sidoporous or pseudoporous. For example, species of Pseudomonas are known to secrete uh, pecalin and pyridine that bind iron. Enterobactin, produced by E. coli bacteria, is the strongest chelating agent known. The marine mussels used metal chelation, especially Fe+3 chelation with the DOPA residue in mussel food protein 1 to improve the strength of the threads that it uses to secrete itself to surfaces. In geology, the application of chelating compound. In earth science, hot chemical withdrawing is attributed to organic chelating agent like peptides and sugar, the exact metal ions from minerals and rocks. Some metal complexes in the environment and in nature are not found in some form of chelating ring with a humic acid or a protein. Thus, metal chelates are relevant to the metabolization of metal in the soil, the uptake and the accumulation of metals into plants and microorganisms. Selective chelation of a heavy metals is relevant to bioremediation, as for example removal of 137 cesium from radioactive waste. We can use the selective chelation process. Medical application as a nutritional supplements. In 1960s, scientists developed the concept of chelating a metal ion prior to feeding the elements to the animal. They believed that this would create a neutral compound, protecting the mineral from being complexed with insoluble salt within the stomach, which would render the metal unavailable for adsorption. Medical applications in which amino acid being effective metal binders were chosen as the prospective ligands, and research was conducted on the metal amino acid combinations. The research supported that metal amino acid chelates were able to enhance mineral adsorption. During this period, synthetic chelates such as ethylene diamine tetraacetic acid like EDTA were being developed. These applied the same concept of chelation and did create chelated complexes, but these synthetics were too stable and not normally viable. Now, medical application and nutritional supplements. If the mineral was taken from EDTA ligand, the ligand could not be used by the body and would be expelled. During the expulsion process, the EDTA ligand randomly chelated and stripped another mineral from the body. According to the Association of American Feed Control Official AAFCO, a metal amino acid chelate is defined as the product resulting from the reaction of a metal ion from a soluble metal salt with a mole ratio of 1 to 3, preferably 2 moles of amino acids. The average weight of the hydrolyzed amino acid must be approximately 150 and the resulting molecular weight of the chelate must not exceed 800 Da. Ferrous biglycinate is an example of one of these compounds that has been developed for human nutrition. dental and oral application. For generation, dentin adhesives were first designed and produced in 1950s. These systems were based on a monomer chelate with calcium on the surface of tooth and generated very weak water resistance chemical bonding 2 to 3 MPa. Heavy metal for detoxification. Chelation therapy is used as antidotes for poisoning by mercury, arsenic or lead. Chelating agents convert these metal ions into a chemically and biochemically inert form that can be excreted. Chelation using calcium disodium EDTA has been approved by the US Food and Drug Administration, that is FDA, for serious cases of lead poisoning. It is not approved for treating heavy metal toxicity. Although beneficial in cases of serious lead poisoning, use of disodium EDTA instead of calcium disodium EDTA has resulted in fatalities due to hypocalcemia. Disodium EDTA is not approved by FDA for any use, and all FDA approved chelation therapy products require a prescription, mandatory. Now, pharmaceutical of chelation. Chelate complex of gadolinium are often used as contrast agents in MRI scan. Although iron particles and manganese chelates complexes have also been explored. Bifunctional chelate complex of zirconium, gallium, fluorine, copper, yttrium, bromine or iodine are often used for conjugation to monoclonal antibodies for use in antibody-based PET imaging. These chelate complexes often employ the use of hexadentate ligand such as diferrooxamine BDFO. According to Mezes et al., and the gadolinium complexes often employed the uses of octadentate ligands such as DTPA, according to Desrux et al. Uranophane, a chelate complex of gold, is used in the treatment of rheumatoid arthritis, and penicillin amine, which forms chelate complexes of copper, is used in the treatment of Wilson's disease and cystinuria, as well as refractory rheumatoid arthritis as well. Other medicinal application. Chelation in the initial intestinal tract is a cause of numerous interaction between drugs and metal ions, also known as minerals in nutrition. As example, antibiotic drug of the tetracycline and quinolone families are chelators of Fe+2, Ca+2 and Mg+2 ions. EDTA, which binds to calcium, is used to alleviate the hypercalcemia that often results from bad keratopathy. The calcium may then be removed from the cornea, allowing for some increase in clarity of vision for the patient. Gold salt, sodium orothiolate or oranophine. This is the gold complex which is used in rheumatoid arthritis. Gold salts are ionic chemical compounds of gold. The term a misnomer is a synonyms for the gold compounds used in medicine. Chrysotherapy and uh, urotherapy are the applications of gold compounds to medicine. Gold salt treatments began in 1935 primarily to reduce the inflammation and to slow disease progression in patients with rheumatoid arthritis. The use of gold compounds has decreased since the 1980s because of the numerous side effects and monitoring requirements, limited efficacy, and very low or slow onset of action. Most chemical compounds of gold are example of metal thiolate complexes, which already we have seen in the previous slide. Now use in rheumatoid arthritis, the gold complexes. Investigation of medical application of gold salts began at the end of the 19th century when gold cyanide demonstrated efficacy in treatment Mycobacterium tuberculosis in vitro. Indications: the use of injected gold salts is indicated for rheumatoid arthritis. Its uses have diminished with the advent of newer compounds such as methotrexate and because of the numerous side effects of the gold compound. The use in rheumatoid arthritis efficacy of orally administered gold is very limited than injecting the gold compounds. Now administration. Gold salts for rheumatoid arthritis are administered by intramuscular injection but can also be administered orally, yet the efficacy is even slower. Regular urine tests to check for protein indicating kidney damage and blood tests are required although. Now efficacy, which is as we found it is little slower. A 1997 review, that is Soares Almazo Me et al. Reports that the treatment with intramuscular gold, that is parenteral gold, reduced the disease activity and joint inflammation. Gold salts uh, taken by mouth are less effective than by injection. 3 to 6 months are often required before gold treatment noticeably improves the symptoms. Side effect. A noticeable side effect of gold-based therapy is skin discoloration in shades of mauve to a purplish dark gray when exposed to sunlight. Skin discoloration occurs when gold salts are taken on a regular basis over a long period of time. Excessive intake of gold salts while undergoing chrysotherapy results through complex redox process in the saturation by relatively stable gold compounds of skin tissue and organs as well as the teeth and ocular tissue in extreme cases in a condition known as chirasiasis. This condition is similar to ergeria, which is caused by exposure to silver salt and colloidal silver. Chirasiasis can ultimately lead to acute renal failure such as tubular necrosis, nephrosis, glomerulitis, severe heart condition, and hematologic complications, that is leukemia or anemia. While some side effect can be healed with moderate success, the skin discoloration is considered permanent. Other side effects of gold salt include kidney damage, itching, rashes, and ulceration of the mouth, tongue, and pharynx. Approximately 35% of patients discontinue the use of gold salt because of these side effects. Kidney function must be monitored continuously while taking the gold salts. Types: Oranophine, that is used in UK and US, Oruthioglucose, that is gold thioglucose, used in US, disodium orothiomylate, sodium orothiosulfate, that is gold sodium sulfate, gold sodium thiosulfate, sodium oronothiomelate, that is gold sodium thiomelate, used in UK. This is enough from my end. Thank you for paying attention to this video lecture. Keep asking questions, as you know that asking question is a beautiful way to gain knowledge. Thank you.